Hello, I'm Mr. Fisher, and this week our asynchronous video will be covering genetics and heredity. We'll be identifying some of the genetic characteristics that are inherited in predictable patterns. In our lesson, we will learn how to predict the kinds of characteristics that show up in offspring and that show up through tools like the Punnett square and the mono and dihybrid crosses. So sit back and let's get started. It's when it comes to genetics, we all know that it takes two parents to get things going and started. But after that, it seems pretty complicated at times. Have you ever wondered why a cute puppy is the only one in the litter that looks like its mother? Or maybe there are others in the litter that look different? Or why not no one in a big family like the one down the street has red hair, except for one person? Many of the mechanisms of inheritance can be complex, but there are some characteristics that can be followed from one generation to the next in a fairly straightforward way. In 1865, long before modern understanding of genetics and DNA, a scientist named Gregor Mendel discovered some of the secrets of inheritance and became the first to describe genetic crosses. A cross refers to the process of fertilization and production of a new generation. And the prefix mono means singular. Di means two. So those terms will become more prevalent as we move on in the lesson. Mendel started off by focusing on his studies on a singular inherited trait or characteristic from the crosses of his pea plant. Gregor Mendel raised hundreds of pea plants for his experiments, and he learned a lot about how they possess or pass on traits from one generation to another. He noticed that for some traits, like the flower color or seed shape, different plants would show only two forms of that trait. For example, plants had either white or purple colored flowers, or rounded or wrinkled seeds. He could also produce a pure line of plants that would only produce offspring with one form of a single trait, so that purple flowered plants always had purple flowered offspring. This was important because when we think about this term, we call it a hybrid. A hybrid then is a plant that is the result of a cross between two plants that are both pure for the same form of the same trait. Gardeners do this all the time, and most of us are familiar with the term hybrid from the beautiful colors produced in hybrid roses. Gregor Mendel's hybrids didn't act the way that the roses do that we know of them. There was no bleeding of colors or seed type. In fact, the offspring of these pure crosses always were 100% of the time looked only like one parent of the plant, never the other. So Mendel decided to find out what happened in the next generation if he crossed those plants with each other, creating monohybrid crosses. Dominant and recessive traits became something that Mendel really focused on in his work. He noticed that one form of the trait occurred more often in nature. This was the form that always appeared in his first generation of his hybrid crosses. He called this the dominant form of the trait. The other was called the recessive form of the trait, as it seemed to disappear from some generations or recede. Although the first crosses of the only two pure strains resulted in all the offspring showing the dominant form of the trait, the second generation, or the monohybrid cross, already resulted in 3-to-1 ratio of dominant to recessive offspring. And we'll look at this a little bit further when we look at our Punnett squares and mono and dihybrid crosses. Mendel's ratios were consistent because he used such large numbers of plants. Recall, this is what happens when you flip a coin. So if we flip the coin, you may not result in 50-50 for the first few times, but if you do it a lot of times, you will come closer and closer to that 50% number. This is what happened when he crossed, and he determined that he could get the 3-to-1 ratio over and over again. Now, all this happened before our modern knowledge of how DNA works and how we pass along information to every cell in every living organism. Because all living things have two parents, it made sense to Gregor Mendel that at least two copies of the information were present for each particular trait. And this turned out to be true. We now call this the DNA information for a single trait, a gene, and the two copies of the gene, which are inherited from each parent, called alleles. For traits that are inherited according to this model, organisms can have three possible sets of genetic information. Either two of them have the same allele for one form of the trait, or one of each of the two different possible alleles. Today, we know that every allele that is inherited is in either way dominant or recessive. If you have received alleles for the dominant form of the trait from your parents, then you will express or display the characteristics of the dominant form. This was like the pure purple plants in Gregor's experiment. If you have both alleles for the recessive form, then the recessive form of the trait will be expressed, like the pure white flower peas in his experiments. The description or understanding that an allele of an organism for any gene is called the genotype, and the word phenotypes describes the physical expression of the two forms of the trait that the gene is expressed or displayed in the individual. To basically say it is what you see. To take a step back, a gene is a small piece or section of DNA that codes for a specific trait. Each gene within itself has a set of instructions for making the molecules that organisms need to survive.
Now life is the oldest story ever told. It's noticed from warmest climates to the coldest. But genes are the code that let it unfold. They determine our traits from our toes to our noses. Genes are composed of DNA, so we find DNA on the chromosomes in every living cell that an organism owns. So genes make a frog from a tadpole. It's a program like Microsoft Word, and it lets your body know just how to grow up. From your eyes to the skin on your arms and your legs, you get it from your parents, from the sperm and the egg. Watson and Crick were on some awesomeness. Life and the double helix causing it. Identical twins have the same DNA, but 98% is the same as a grade 8. But it's those little differences that make us unique. You can try to make it the same with the cloning technique. I get so smooth on the beat that you might have to rewind it. Not your Levi's kid. These genes are for life.
I said we are DNA. Don't stop, we don't play. Like genes control your face, like dominant traits, we dominate. Hey, we are DNA. Don't stop, we don't play. Like genes control your face, like dominant traits, we dominate. Hey.
Traits aren't just random, they get passed along. Like you probably won't give birth to a mastodon unless you happen to be a mastodon. But if you're listening to this song, I guess you probably aren't. Traits get passed to you from your parents, kid. From sexual reproduction, yes they did. If your mom is blonde and your dad is dark hair, you're like, why am I not blonde like my mom? The blonde genes you got from your mom aren't strong. Dark hair's a dominant trait, it dominates. But the blonde genes from your mom aren't really bad, they're just recessive, they hide it in the back. One time I cut my thigh, I was like, this is hopeless, I think I'm gonna die. I forgot about mitosis. Cells divide and grow into new. Skin comes back like a boomerang. Who are you?
I said we are DNA. Don't stop, we don't play. Like genes control your face, like dominant traits, we dominate. Hey, we are DNA. Don't stop, we don't play. Like genes control your face, like dominant traits, we dominate.
Genes are units of heredity that are identified as different versions and can have different versions, which we call alleles. A gene usually has two alleles, one from the mother and one from the father. Each allele is assigned a letter when we talk about genetics, either an uppercase or a lowercase letter, indicating whether it's recessive or dominant for a particular trait. For example, an allele for the patterning of eye color may be represented by a C if it is dominant, or a little c if it is recessive. But what happens if an organism gets one dominant allele and one recessive allele? The dominant allele, as the term dominant, will overpower it or mask that allele. However, if an organism inherits two recessive alleles, then the recessive pattern will show, or the recessive trait will show. If an individual has the same two alleles for a gene, either big C or either two dominant alleles In that case, it will be called homozygous. If it has two recessive genes, it will be called homozygous. If an individual has alleles or has different alleles, let's say a dominant capital letter and a recessive lowercase letter, we call this heterozygous. You can remember this by the homo means the same and hetero means different. The combination of alleles of an individual inherits is its genotype, while the physical manifestation of the gene or what the individual looks like or what that trait presents is known as the phenotype. To determine all the possible genotypes and phenotypes of an offspring, scientists perform crosses to predict the outcomes. To do this, we use a tool known as a Punnett square. This shows the types of genotypes of a cross by using the letter representations of the different alleles. Capital letters represent dominant alleles, and lowercase letters represent recessive alleles. The top rows show the two alleles of one parent. The left column shows the two alleles of the other parent. And the four boxes, when we look at a monohybrid cross, with the two letters, illustrates only the possible genotypes of the offspring. A monohybrid cross is the creation of a new generation from two organisms that are hybrids from a single characteristic, or we're looking at a single trait. Mono meaning one, we're looking at one single trait. They become hybrids because the parents that contain these traits, we predict the possible outcomes by combining their traits with each other. These monohybrid parents can pass on their traits in either allele from each generation, and the different combinations results in the possible types of genetic information or genotypes. You can either have two dominant alleles, two recessive alleles, or one of each in each of the boxes. A dihybrid cross is a cross between two individuals that are both heterozygous for different traits. These crosses show up even though the parents have the same genotype or genetic makeup, or phenotype or physical appearance. Their offspring could have any of 9 different phenotypes, 9 different genotypes, 4 different phenotypes. And just like in a monohybrid cross, each parent contributes one allele for each gene, and every parent allele has an equal chance of being given to the offspring. To represent this accurately, in this we set up our dihybrid crosses, we use the FOIL method. FOIL stands for first, outer, inner, and last. And we use our parental genotypes to set up the top lines and sides of our Punnett square. Once completed, we can compare the genotypic and phenotypic ratios, just like in our smaller monohybrid crosses.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase Y always overpowers its lowercase friend, so the only time you get green babies is if you have two lowercase y's.
In Mendel's first generation, the yellow homozygous P mom will give each P kid a yellow dominant allele, and the green homozygous P dad will give a green recessive allele. So all the P kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
We call the traits that we can easily compare using monohybrid and dihybrid crosses Mendelian traits, and these traits are passed down on one gene with dominant and recessive alleles. The phenotype for the recessive alleles is only shown when an individual has two recessive alleles from both parents. However, when there are non-Mendelian traits, such as hair color, that don't follow the simple pattern of dominant and recessive alleles, we say that they are non-Mendelian. They follow non-Mendelian genetics. Non-Mendelian traits can be categorized as polygenic, codominant, incomplete dominant, sex-linked, and there are a few different more, but those are the ones we typically look at for in our level of biology. Polygenic traits are considered non-Mendelian traits because the alleles are located on more than one gene, which allows for more alleles and phenotypes. Examples of polygenic traits are hair color and height. Other traits, such as blood type, show codominance, where there is no dominant or recessive allele. In this case, both alleles are expressed completely. So an individual has both alleles will show it with both phenotypes. This is why individuals with AB blood types will have both A and B markers in their red blood cells. Nose size, ironically, is a trait controlled by incomplete dominance, where neither allele shows complete dominance. This is different from codominance because instead of showing both phenotypes, traits controlled by incomplete dominance will show a blended phenotype that is somewhere in between the two phenotypes. For example, individuals who have one allele for a large nose and one allele for a small nose will show a blended phenotype of a medium-sized nose. Other traits, such as red-green color blindness and baldness, are sex-linked. Alleles for these traits are passed along to children on the X and Y chromosomes. DNA that is responsible for gender, often with these alleles, are only found on the X chromosome, which means males are more likely than females to be bald or have red-green color blindness. Now, I know this was a lot of information for today. Please take your time, rewatch the video as many times as you need. Thank you so much for your time. And I look forward to seeing you in our live sessions this week. Have a wonderful day.